Okay, wonderful. Welcome everyone to the last seminar of the 2021 year. It's hard to believe it's already December, but we're really happy to have you here. And we're excited for the talk we have today. Next slide. First, I'm going to go over a few housekeeping items. You should have received a notification when you signed in. This seminar is being recorded. All participants are muted. However, we still welcome you to ask questions and make comments. You can put those into the chat function, and we will administer those to our speaker at the end of the presentation. Spanish interpretation is available, and you can use that by selecting either English or Spanish from the interpretation language selection below. It looks like a globe in your lower menu bar on Zoom if you haven't already selected it. And then closed captioning is also available, and you can select the CC icon below and then click Show Subtitle. Great, thank you so much. So I'm really happy to introduce our speaker for today. Dr. Parikh is a professor in the Department of Land, Air, and Water Resources. We have had the opportunity to fund a couple of his research projects, and we're excited to hear him speak today about the potential impact of nano-based agrochemicals, biochar soil amendment, and wildfire ash on farm worker safety. So Dr. Parikh, I'm happy to turn it over to you now.
All right, thank you so much for the introduction. I'm going to go ahead and share my slides. Okay, I'm all set now. So, all right, well, I appreciate being given the opportunity to come here today and share research that I've been doing with my group over the last many years actually. I'm currently funded through the Western Center for Ag and Health Safety to do work on the potential impact of nano-based agrochemicals. However, they have previously funded me to look at similar studies looking at the farm worker safety for biochar and wildfire ash. And although I could have just gone ahead with as planned, just the nano-based agrochemicals, I thought that going into such minute detail may not be as interesting as me kind of pulling these all three things together, as they are certainly related. So in beginning, I'd like to thank again funding from the center, and we have some contributors that I just want to acknowledge. The work for the nanoparticles primarily done by Janice Patino, for the biochar dust by Dr. Lee, and for the wildfire ash by Dr. Wong. So certainly there are concerns, soil contaminants, and there's different pathways at which those soil contaminants can come into humans and cause concern. And one of the most obvious would be food and water. So certainly water contamination is something we know about. In Davis even, we had hexavalent chromium in our groundwater, and that's why we now use Sacramento River water for our drinking water. And so whenever food is grown in soils that are contaminated, it does threaten food safety and security. And these contaminants that are in the soil or in the water can accumulate into crops. So those crops can carry those contaminants to your dinner plate. Also of concern is soil dust. And there's been a growing interest in the potential negative impacts of soil dust. And that's just talking about dust in general, because dust has small particles that can be harmful. Particularly, we're talking about the PM 2.5 and PM particulate matter of 2.5 microns and particulate matter of 10 microns. But if those particles happen to be carrying any contaminants on them, those contaminants are also going to be inhaled and could carry additional risks. And so these projects are aiming to look at that and make an assessment of what that risk really is. So Going to start with agrochemicals, then swing into biochar, and wrap it up with wildfire ash. So nanoparticles are actually everywhere, and many people are aware of this, but they're commonly found in like sunscreens, cosmetics, other personal care products. They're used in paints and in electronics. Our electronics actually have a lot of nanoparticles in them. And these nanoparticles are not going to be contained. There is a release. And so when that release occurs, they enter the environment, and they can enter the aquatic system, our soil systems, or even enter the foods that we eat. So people are wondering, is this a problem? Like, how much of a problem are nanoparticles in the environment? And, you know, they can be toxic, but how toxic are they? And for a long time, there was a great deal of concern that using nanoparticles was going to be a big problem for food safety, and it would accumulate in the food and have negative effects on people and animals. However, over time, we've found that that doesn't really, the threat isn't nearly as great as we had initially thought. In aquatic systems, there tends to be more of a concern and more deleterious effects. But for agriculture, it's not as much as a problem. The reason for that is when we release those nanoparticles, or they get released accidentally, they're in a dispersed mixture, and they're very small particles. And these small particles can have some of those toxic effects. However, in the natural systems, they start to come together and aggregate or agglomerate. And so they don't behave as small particles, they actually behave as larger particles. And so this aggregation process reduces the potential toxicity. And so because of this, people have started to ask the question, what if we use nanoparticles to benefit agriculture? And that would be, can we use nanoparticles as a way to deliver fertilizers or to deliver pesticides to agricultural fields? And the aim being here that we have improved crop productivity and high efficacy requiring less dosage and therefore reduce cost. And also as a way to have materials that have a longer shelf life and persistence. Now, these nanopesticides or nanofertilizers or nanoagrochemicals are released to the environment. That's their job, that's their purpose. And so it clearly is a potential pathway for worker exposure. The thing that's most concerned is the inhalation of the PM2.5 and PM10 material. And that would be during the nanoagrochemical application, so how they're applied, and then through agricultural dust, which could be from wind or tillage operations. Just to get the sense of how big these particles are, this is a human hair, and this is the cross section of that. This is the PM10, and this is PM2.5. So these are really small particles. So we went and obtained a number of these commercially available nanoagrochemicals, and the ones we used were Coside, Quavo, and NanoPro and NanoRise. And we then simulated dust generation, and I'll get to how we did that. And then we tested the toxicity of the nanoparticles themselves and the nanoparticles in these dust fractions. And in addition to that, we quantified the amount of the nanoparticle from their metal per point of view, in this case, copper or silver, using ICP-MS, which is this fancy instrument over here. So in the dust generation process, this was actually developed by Dr. Randy Southard, who retired recently from UC Davis. And the idea is that there's this rotating chamber here that is, at a specific rate, and there's going to be, various moisture contents that are used, and a blower that blows the material into the second chamber, the settling chamber. And the larger particles will settle on the bottom, and the small particles remain in the air fraction and can be collected by vacuum pumps that are pulling on these impactors that collect those size fractions of dust. For these experiments, we used a local soil, the Yolo silt loam, The groundwater that we used for adding the moisture to the soils was from Campbell Tract in Davis. And then we have the nanoparticles I previously stated. We also added some silver colloids. When we look at the properties of the nanoparticles, it's really not important for us right now at this stage to go through all the details. But this is the zeta potential, and this represents the charge of the particles. And we see that they all have a negative zeta potential. And that may end up being important later on. Of course, we see copper in them, and we see silver in the silver particles. The groundwater has very trace levels of these. So the way we tested for potential toxicity is a bioassay called CALX. And that's the chemical activated luciferase gene expression. And so this tests for potential endocrine disrupting chemicals using modified human cell lines. And so if the chemicals present in our sample, in this case, the nanoparticles of the dust, have potential to be in endocrine disrupting chemicals, then there's going to be a light emission. So the compound comes in, if it binds to the receptor that we're testing, and it will then have this light emission. And so you have this process occurring in a little tube, and then we can easily read the positive response, and the intensity of that response is also important. So we use a few different targeted receptors here, the aryl hydrocarbon, estrogen, and androgen receptors. And so these would all be good tests to see if there's a potential for disruption to the endocrine system. Now, when we here's the data from the dust particles and the nanoparticles on themselves. The first thing is to say the nanoagrochemicals do not show potential endocrine disrupting activity. And so those are these over here on this side. And then you see when we have D, that means the dust with those. And so clearly for the AHR receptor over here, the aryl hydrocarbon, there's a negative response compared to the percent of TCDD, which is essentially the standard to show a response. And then over here, when we have the dust present, we see that those actually become slightly positive, but those numbers are low, so they're not of concern. And so the soil itself has is giving some response in this case. For the estrogen receptor, we see that pretty much it's negative across the board. And then for the androgen receptor, there's it's higher than the AHR positive response. A lot of it coming from NanoPro and NanoRise, which actually are solutions that have fulvic acids in them and are meant to be fertilizing materials. And so it could be that those materials are actually responsible for that slight increase. When we look at the total concentration in the dust of the metals, we can compare here, this is copper on the left, the amount of copper in our treatments. Over here is just groundwater. So this is the response for, or the amount of copper measured in the different size fractions. And definitely, as we go use these nanoparticles, we're adding copper to the soil, and the concentrations in the soil do increase. And for some reason, this copper plus nanopro has a very high amount of release. Comparing the silver, when the silver colloids are used, there is an increase. So while these metals can have some toxic effects, when these metals were present in higher concentration, there was no observed risk using the CALX bioassays. But one thing that was particularly interesting is that the application of these NACs led to greater dust emissions. So when we compare the groundwater only, we have the dust emissions here for 2.5 and 10. And as we look across, we see that for the 2.5, many of them are higher. And same for the 10 micron, many of them are higher. So something about adding these nanoparticles is actually causing more dust to be released. We don't see that there's a real negative impact from our bioassays, but we believe that because these particles are negatively charged, Soils tend to be negatively charged as well, that they could actually be getting in there and causing some dispersion of the soil aggregates. And so they're less stable and more likely to break apart and enter into the dust fraction. So as we conclude this section from agrochemicals, what we can say is that according to our tests, the analyzed products do not contain endocrine disruptive compounds. And if you follow the application label, which is what we did, so that's the manufacturer's intended dosage, there is agricultural dust, but it's not greater than the soil alone. And this is in spite of the increased concentrations of the metals, copper, and silver. The dust generation did increase, and therefore it means there's a higher probability of inhalation of the dust. We also are now looking at what happens if someone doesn't follow the label, and so we increase the dosage a factor of 10. This was actually recommended to us by one of the nanoparticle companies that we are working with, because he said that people don't follow the label, and they wanted to know what would happen if someone did increase the concentration. So moving on to biochar. So we need to figure out what biochar is if we don't know. And biochar is a category of charcoal-like materials that have been created through the thermochemical conversion of waste biomass like wood or grass and nutshells. And this is done under high temperature and limited oxygen. And it's being used and promoted as a soil amendment for agricultural and environmental applications. And so why did we come up with biochar? Well, it goes back to a discovery, so to speak, of the Terra Preta de Indio soils, or Amazonian black earth, in Brazil and South America. And it was determined that these deposits of charcoal are 500 to 2,500 years old. And when we compare the difference of the similar soil having had the charcoal addition and not having it, we see these dramatic and impressive results for the conditions that they're showing here. So biochars are really different, though. It's important to remember that. So if someone's going to apply biochar, your question should be, oh, what kind of biochar are you applying? And so it's a function of the feedstock and the temperature at which they're produced. And this impacts their elemental composition, pH, porosity, surface area, ash content, and water holding capacity. And as I mentioned, it's a function of the production temperature, method, and feedstock. So we have varied products, so we shouldn't expect them to act the same. They have varied outcomes. Now, here we see just that there's various ways of making biochar. Traditional way is just by using earth, soil to bury it to prevent the oxygen. There's the industrial scale where you can make large capacity. And then there's the interesting portable scale where the pyrolyzer unit can be brought to the farm, and then the waste can be converted to biochar and put right on site, which reduces a lot of transport, which is really important. I'm not going to get into the yields here. I don't think that's important for this talk. So when we think about what can biochar do and what are the potential benefits, the first thing is that it's climate change mitigation strategy. So the biochar itself is a recalcitrant form of carbon. It's not going to be respired as rapidly. And so instead of going as carbon dioxide, a greenhouse gas, it will be stored in the soil for longer. It's used a lot in agriculture because of the potential benefits to improve yields, increase soil fertility, and increase water savings. It's also used in remediation because if you think activated carbon, right, that's a similar material. It's not the same, but it's similar. And so these have a high capacity to bind contaminants, so both organic and inorganic. There's also been a lot of use in product substitution. And so there are people who are using this in feed and bedding materials or potting materials. And then we can get these two other sort of added benefits, and that certainly waste management is a problem. Biomass is a problem in California in terms of waste. So it's something that could be used for that. Also, it can be produced in order to generate some bioenergy. And so that is another potential benefit. But even though this talk is not about what are the impacts or results of using biochar, I just want to be sure that everyone knows that these benefits are inconsistent. They're not always realized. There's multiple variables and possible drawbacks. So we find that biochar can help, but it doesn't help all the time, say, for agronomic purposes. So biochar has certainly seen a rapid growth in interest. So starting from 2000 to 2020, we see these purple bars, which indicate the scientific publications. And so it's growing at a near exponential rate. US patent applications are also growing. And so there is this increase of people studying this, and there's an increase of people wanting the information, and there's an increase of people wanting to use this material. Biochar is available in many forms for your home garden. It's also available on the farm scale, so you can have large amounts of these delivered and used on site. So my research group has studied many aspects of biochar, from agricultural uses and environmental remediation. And I've also am very interested in the potential health and safety effects of biochar, and that's what we're going to get into now. So I first want to say that safe use of biochar can be easily achieved. However, as with any product, the proper safety precautions should be taken. All right, so the question is, what is the potential for biochar to become part of agricultural dust? And does biochar pose a greater toxicity risk than soil-only dust? And so there's been some examples. Here's a nice figure from a paper by Ravi et al., where they show how the biochars can break up and there can be this increase of particulate matter emissions. And here's some very irresponsible application of biochar, and it's not going on the field as much as it's going into the air. So there's been prior studies that have shown that biochars can have polycyclic aromatic hydrocarbons and dioxins or other bound contaminants, and that those bound contaminants can conceivably be taken up by plants or enter other pathways for human exposure. And so there are some, but not all biochars that have these elevated compounds of concern. In prior work we did, we wanted to know, well, what if we use biochar in growing, in this case, lettuce? And what we saw was that this walnut shell biochar we were using actually stimulated the aryl hydrocarbon receptor. So it had this potential endocrine disrupting behavior. And so it suggested the biochar itself had some chemicals present, such as PAHs or dioxins, that were causing this. And even the plants themselves, the lettuce and carrots we grew when biochar was present, also showed some of this AHR activity response. So we used the same dust simulator that was used for the nano agrochemicals. We used the same yellow silt loam. We also used the sandy loam. We used this walnut shell biochar I mentioned, and then we used a pinewood biochar produced at 3 different temperatures. Now this is the mass concentration of PM10 in the dust. This is different moisture contents, and not surprising, absolutely as expected, the amount of dust decreases as you increase the soil moisture, and the amount of dust increases as you increase the amount of biochar put in. But these are obviously important things to bear in mind. One thing that was, really surprising or interesting is that when we look at the luciferase activity, so this is that potential negative response, this walnut shell biochar had pretty high activity. And we compare that to the pinewood biochars, and they have very low, almost basically the same as no char. And these are produced at 900, and this one's at 900. So there's something very unique about this walnut shell. And not surprisingly, as you increase the dosage of that walnut shell biochar, the response of the luciferase activity increases. So the walnut shell is something of concern. Most biochars are not problematic, I'll say that now, and this walnut shell biochar that we used, the producer became aware of this problem and was able to fix it. So things can be done properly. We just have to make sure we are. So when we want to apply biochar, some safe some considerations for safety must be followed. The first thing is select the right biochar. Test it in advance. Don't use biochar made from construction debris. Use biochar of a high moisture content. Over here, we have a biochar of a 60% moisture content. There's no dust when it's being applied compared to this one over here. Apply when wind levels are low, and you can actually put the biochar in bands and bury it so that it would be less at risk for wind erosion. This is that process, but you can see that while it was exposed, there's this little dirt devil of biochar. And of course, use the appropriate personal protective equipment. So finally, we move on to the third part, and wildfire ash is different than biochar, of course, but it is still a thermochemical conversion of biomass. And we get ash and not biochar, and they definitely have different properties. But our question was, what happens to this ash when, say, a orchard burns down and the cleanup has to happen? Is that ash going to expose any of the farm workers or other workers doing the cleanup? Is it going to expose them to potentially toxic compounds? So our study was on the Thomas Fire, which occurred in 2017 in Ventura and Santa Barbara counties in California. It was a big fire, burned over 280,000 acres. And we can see the location of where the fire occurred. And then these are actually our sampling locations for our study. And here's an example of the extent of some of the burn, and that these orchards did get impacted. So we studied 3 burning levels, which are slight, moderate, and heavy. And we were doing these in different orchards or plantations of avocado, lemon, and orange. Now, jumping right into the data of the metals present, we see that the levels of the arsenic, this should have been cadmium, arsenic, cadmium, chromium, copper, nickel, lead, and zinc are all higher in the ash than in the soil. So these boxes are the mean, and so this is the ash and that's the soil. So we can always see that those are higher in the ash than the soil. So the ash is being concentrated with metals because whatever was in the biomass that burned, that the organic material can be released as carbon dioxide, but the mineral components and the metals will remain. So that's why the ash has these higher contents. And so obviously that means that cleaning up ash, we should be more careful than working with soil alone. As far as mercury goes, mercury actually volatilizes, and so a higher temperature led to lower concentration than in the soil. Now, there was one site where there was some structure that burned. And on that site, the concentrations of copper, chromium, and arsenic were really high. And so those 3 happen to go into chromated copper arsenate, which is a wood preservative that has been used extensively. And so it just shows that what burned also makes a really big difference in terms of the potential toxicity and any approach to cleaning it up. And so we saw that in the ash, there was higher concentrations, but also with the soil for at least the chromium and copper. So the soil underneath was impacted. We went ahead and did, took the soil samples back to the lab and did these dust simulations as before. And we collected the soil and ash dust fractions of the PM10 particulate matter. And then we used the EPA method to do a risk assessment. And so we don't need to get into all of this, except know that we had the concentration of toxic elements. So those are what we determined. And then the particle emission factor, which is the value obtained from the dust generator itself. And then we used all of this to determine a hazard quotient, which is a way of indicating the level of risk of human exposure. So when we did this, all elements except mercury, the hazard quotient value was higher for ash samples than soils, not at all surprising. But most of them were less than 1, and that 1 is about the cutoff. So if it's less than 1, it's usually considered acceptable. And so although there were higher levels of all these metals, they didn't really raise alarms in terms of how EPA would evaluate the hazard of this ash material. Certainly the particulate size is different, right? And so, but this is just from the toxic compounds within it. But for that site that had that elevated arsenic, copper, and chromium, the HQ values were pretty high in some cases. And we see that in the ash, for all of them, it's above 1, but the soil beneath it also was above 1 for arsenic and chromium. So again, what burns makes a really big difference in terms of how cleanup should be undertaken. So compared to soil alone, wildfire ash poses a higher occupational risk during on-site cleanup, but it's really of concern when we're looking at residential or industrial areas that may contain higher toxic elements. And so like the wood that we observe. And so when we have various sheds or structures and barns that burn, those could be areas to pay particular attention to. We also, I just want to thank Ben Farber from the UC Cooperative Extension Service who assisted Dr. Wan with the soil sampling. So as we kind of bring this all together and wrap it up, just to kind of go over all the results here, is that proper safety measures must be taken. And so when you're using materials or we use cleanup, you should dress like this. These materials, biochar, nanoparticles, and ash, are something to be concerned about, and one should take appropriate precautions. The initial evaluation of these nano agrochemicals didn't indicate that the present was greater risk than soil dust alone, but they did increase dust emissions, and those dust emissions did have higher levels of those metals, but not particularly high levels. They're sort of moderately low levels. And there are biochars that present health risks, but we need to know that there are safe biochars. So if you wanted to use biochar and a producer was going to sell you some, you should first say, what was it made from? What were the conditions it was made? And let me see the lab reports. And so that most biochar producers have those. And if they don't give it to you, then you shouldn't use it. Also, the last thing was for the wildfire ash, that the ash concentrates the toxic compounds and definitely poses a greater risk than soil. So additional safety measures, have to be taken when doing cleanup in these areas, and particularly when structures are burned or additional toxins may be present. So with that, I'd like to thank you for your attention, and I'll be glad to take any questions.
Wonderful. Thank you so much. I appreciate you combining all the different topics because they are all very interesting, and we do have a handful of questions. I actually am going to pick up close to where you ended, which was about the biochar being able to be made more safe. And so one question has to do with what did the walnut char producer do in order to make it more safe? What's that process?
Yeah. So the reason that it in this case that it had elevated levels of these polycyclic aromatic hydrocarbons is because after it was done in a chamber unit. And so after the conversion occurred, the material sat there and the gases above condensed down, and they brought those chemicals with them. And so there's other systems that are flow through, and those would avoid that right away. But in this case, they actually had a pretty ingenious solution. They took biochar that they made and created a filter for their gases. And so they were able to remove those chemicals from the air, and therefore their product was low in these compounds, but they created an incredibly toxic byproduct, which obviously they'd have to dispose of.
Got it. Interesting. Thank you. So you said that an operator could ask for details about the char that they're purchasing. What likelihood do you think that many of those operators are doing that? Is there knowledge in the industry that that's a required step?
Yeah, if you want to be a certified biochar, and you can be certified by the US Biochar Initiative or the International Biochar Initiative or the European Biochar, you have to put that data out there, and that's part of the process of certification. So there is a reason they want to do that. Even people who haven't actually gone through that process, they do have these reports, at least when they talk to me, and they provide them to me. And they usually come from control labs, this one place, and I think they do good work. So there's not a lot of places that do biochar testing. So.
Got it. And then what about, so, one of the audiences of interest for us at the center are farm workers who oftentimes have much less of the safety information or training in how to interpret the safety information. Do you think there's an access point for those workers to learn more about the different materials being used or found in their workplace?
So I guess this is a biochar or all-encompassing sort of all those particles that I've been talking about?
I guess in this case, it would be biochar because it's the one that somebody's intentionally putting down opposed to the wildfire ash.
Well, so certainly asking for a certified biochar is your best bet, because there are threshold values that, they have to be below in order to be certified. And so that will guarantee that you don't have high levels of potentially toxic organics or inorganics. And so that's the best way. If they don't provide you the data, then certainly don't. But if they do give you the data, they will state the levels on it. I actually don't know if they say it's too high on those reports. I don't look at it that way. But I'm sure that you could follow up and ask that question, and contacting the lab that did it, I think they would be very willing to help interpret that report for them.
Great, thanks. So why don't we go towards the nanoparticles, and we have a question from somebody in the audience asking, does mowing cover crops and dry grasses create nanoparticles? So
mowing isn't going to, so the nanoparticles that I've been discussing are metal oxide nanoparticles. And so it's certainly not going to create those, but it will create particulate matter. And some of that particulate matter could be in the nano size range, but you're not going to be producing the so-called engineered nanoparticles or nanoparticles that I've been talking about. There are definitely naturally occurring nanoparticles. Our soils are full of them. When we have iron oxides that are created in soils, they initially start off as nanoparticles, and then they grow. And so there's a lot of natural nanoparticles. The focus is that how do we use these nanoparticles that we synthesize for specific intended outcomes? But as far as the mowing, it's just going to be the typical dust that you would consider.
Great. So a question from Dr. Pinkerton from early in the conversation. He's asking whether or not it's known, or is it known whether nanoproducts, so nanopore, et cetera, are actually nano in size or less than 100 nanometers in diameter?
Yes, it's known, at least to us. And the company Aqua Yields that has NanoRise and Nano Pure, is it? I can't remember, but they gave us all their data. I mean, they gave us everything. And actually the NanoRise doesn't even have nanoparticles in it. It's an interesting one. It's made for people to put nanoparticles in, and then that fertilizing material would be distributed. The other one already has that in there. but they do it so people can use whatever silica nanoparticles, copper, whatever they'd like. The other particles, the Quavo and Coside, we did for all of them actually, the SEM analysis, so microscopy, and determine that they are, and we have the size distributions, but I don't recall them.
Great, thank you. Okay, we do have a couple more questions that I'm going to go through, but feel free to drop additional questions into the chat if you have any. Let's see. Going now to the wildfire ash, so we're aware of wildfire smoke exposure regulations, and employers, and so that's the smoke, employers are supposed to protect workers from any known hazard. So do you think it's known among growers or anyone, I guess growers, farmers, about the potential hazard of the ash material during cleanup, or is that an area where there needs to be more education? It sounds like there's a potential hazard there, especially if there were structures on site.
Yeah, so I don't know. I feel like, you know, obviously people are aware that there's dust, and I know that farmers tend to be very responsible, and they wear their protective equipment when they're applying pesticides and such. So, you know, at least it's not something that I think would be inconceivable. But I don't know that, you know, after what happens when there's a wildfire, and because that's an extreme event, right? And so, do people get that sort of information before they go out and start, you know, moving the debris around? But they should get that information if they're not.
Sure. Well, unfortunately, with the increase in wildfires, that might be an opportunity for future outreach and communication that we can collaborate on. I know your focus is on, you've been talking a lot about the dust and the respiratory health implications, but I'm wondering if there are also potential dermal occupational, you know, hazards that workers may encounter from interacting with these different particles.
Yeah, Bob Rice asked that question. So I've talked to Bob Rice. He's a professor at UC Davis, and we have talked about during doing these dermal exposure studies, and I'd like to do them. And so hopefully we will get to that.
Great. I'm pleased to say that I managed to think up that one, which shows that I'm learning more about the numerous hazards in agricultural health and safety. Okay, we have another question from someone in the group saying, what is your opinion about spraying foliar fertilizers to crops such as iron, manganese, and magnesium? Can the above have the potential to produce nanoparticles?
I think that it's fine. Foliar application can be a really effective way of giving certain nutrients to plants. And, you know, a lot of it will go towards the soil, right? It's not all going to be delivered on the leaves. I don't think that those are going to be really forming nanoparticles, but if they did, even you need to have like a really high concentration in a certain area to form a nanoparticle. And so, yeah, maybe if it fell on the ground, that could happen. But those nanoparticles are not going to be particularly toxic. So, like I said, iron nanoparticles exist in the soil anyway, and because the soil is such a heterogeneous mixture of clay and organic matter and microbes, all of these things kind of work as glue in a way to help pull these particles together. So they don't really exist as as individual nanoparticles. In my area, I don't do any work on foliar application. Well, that's not true. We did look at copper in terms of vineyards a little bit, because in that case, you're using copper as a fungicide, and the soils end up having incredibly high levels of copper, particularly those in Europe where this has been done for so long. But those, in that case, that copper is bound very tightly, and it just happens, so it's not particularly toxic to the plants. But yeah, I don't have like the perfect answer for that question about foliar application, but I wouldn't be concerned about it myself.
Great, thank you. That made me think of another question, which is back to the ash composition and whether or not you think it would vary much by crop or commodity. You know, I think that with the orchards that you looked at, I'm not familiar with the frequency of use of different pesticides or fertilizers as much by commodity, but do you think that there could be some in which that ash composition becomes dangerous?
The ash from repeated fires, is that?
Well, I don't know about repeated. I guess I mean like, you know, the soil with the ash post fire was not dangerous in the study that you, or, you know, not above a threshold in the study that you did in the avocado and citrus orchards. Could it be in a different, and would it vary by commodity potentially?
It shouldn't really value by commodity, but it would vary based on the soil properties and the water source, because the plants are going to be taking up those metals that we've studied essentially through the soil from weathering and those materials being in the soil, or through the water. And so the concentration that is in the plants themselves matters, and that's a function of the environment in which they're grown. Now, certainly some plants do take up more of certain chemicals than others, like spinach is a cadmium hyperaccumulator. And so it's going to have a lot more cadmium in it than a strawberry. But that would be the only way the plant species differs, if it happens to be a hyperaccumulator of a specific metal.
Great, thank you. I think we have gone through all of the questions from the participants, the audience. And so I just want to thank you so much for joining us. And I hope that our center will be able to continue supporting the work that you're doing, because it is very relevant to our great agricultural industry. And I want to thank everybody for joining. We are going to be off until February. We do not have a seminar in January, but we'll be back at 4 p.m. the first Monday of February. So I hope everyone has a wonderful new year and a safe one, and we'll see you in the new year. Bye.